Hello students, so today we are going to learn a new chapter, that is living world and classification of microbes. Now from the name of the chapter living world, in this world there are many different species present, right, from smaller to bigger, that is macro and micro. Macro, which can be easily be seen with our eyes, whereas micro, they are very minute. Some are not visible with our naked eyes, which requires microscope, right. Now these species we are going to learn by classifying them. Few species I added the images over here. You can see how they are moving around or locomoting. Here you can see this species moving with the help of cilia. Further, in this chapter, we are going to learn the introduction part, biodiversity and need of classification, criteria for classification, kingdom Monera, kingdom Protista, kingdom Fungi. Classification of microbes, that is bacteria, protozoa, fungi, algae and viruses. Now students, if you see here, I just added a simple image showing the bacteria present in our hands. That is, whenever we go out and come, it is mandatory to wash our hands. Reason, we may carry many microorganisms, and if you don't wash our hands and have food, these microbes can enter our body and cause various infection. Also, few more animation added over here. I can say this particular animation looks like a beautiful rangoli, right? Also, here we can see amoeba species which is locomoting with the help of the false food, that is pseudopodia, and few microbes moving around, whereas the microorganism feeding on much more smaller organism. Now, coming to the history part of it. Now, Carolus Linnaeus in 1735 divided living world into two kingdoms, Vegetabilia and Animalia, that is the plants and the animals. Further, Ernest Haeckel in 1866 considered three kingdoms, that is Protista, plants and animals. Now, what is this Protista? They are the microorganism which Haeckel had kept in a different kingdom. Then further in 1925, Chitin created two groups, prokaryotes and eukaryotes. In 1938, further, Copeland divided living organisms into four kingdoms, Monera, Protista, plants and animals. Now this prokaryote, eukaryote are based on their presence of cells, the primitive and the present cells. And based on that, Monera and Protista. Now the name of these scientists is Carolus Linnaeus. I added the image so that we can come across their work, who they are. So Linnaeus is also known as the father of modern biology, classification, or the father of taxonomy. Ernest Haeckel, Copeland and Chitin. Moving further, biodiversity and need of classification. All living organisms on Earth have adapted according to geographic regions, food ingestion, defense, etc. Now, what does that mean? The species, they are present in different climatic condition, whether it is water, air, mountain, soil, everywhere they are present, and they adapted accordingly. And food ingestion, again some ingest with the help of any siphon, suckers or directly gulp in, and they have got various defense mechanism. While adapting, many differences are observed in the organisms of same species too. According to 2011 census, around 87 million species of living organisms are found on Earth, including land and sea. So students, you can imagine 87 million species. We can remember 10, 20, 50 or 100. But 87 million species to learn, remember, it will be very difficult. That is why they are classified according to the shape, size, structure, etc. On to the right side, you can see I added around 30-40 images of different species. You can see these are just a few images. To study such a vast number, it was essential to divide them into groups. So groups and subgroups were created considering the similarities and differences among the living organism. So the species were divided into different categories, the similarities, differences, so that they can be easily studied. So groups and subgroups were made. The process of dividing living organism into groups and subgroups is called biological classification. That is how they are classified biologically. Further, Robert Harding Whittaker was an American ecologist. In 1969, he divided living organism into 5 groups. So in the previous slide, students, you saw the different scientists classifying based on their criteria, structure, shape into 2, 3 and 4. But finally, Robert Whittaker divided all these species into 5 groups. For classifying or classification, Whittaker considered following criteria. Now students, to learn these many species, Whittaker had put up certain criteria so that it becomes easier to understand and learn. First is complexity of the cell structure, prokaryotic and eukaryotic. That is, as I mentioned earlier, the oldest cell or the primitive cell, and the eukaryotic, the present known cell. So the based on the complexity of the cell, it is divided into 2 categories, prokaryotic and eukaryotic. Complexity of organism, unicellular, multicellular. There are species which are single cell or many cell. Uni means one, multi, many cellular. Mode of nutrition. Plants which are autotrophic, photosynthetic means autotrophic, the one which prepares their own food. Photosynthetic, that is using the sun's light by the presence of chloroplast in the plants. Fungi, saprophytic, absorption from dead organism. Now these species are the one which feed on dead and decaying organisms. Animals, heterotrophic and ingestive. Now animals, they are dependent on the plants, so they are heterotrophic and they take in the food, that is ingest. Fourth, lifestyle. Plants produces, that is with the help of autotrophic mode of nutrition and photosynthetic, they produce food. Animals, they are consumers, and fungi, they are decomposers, that is saprophytes. Phylogenetic relationship, that is prokaryotic to eukaryotic. Now as we know, the earlier known species, that is right from prokaryotes, the unicellular primitive ones, to the present day modern eukaryotic cells, from unicellular to multicellular. Now students, here I added a simple image. Again, it's a animation one, slightly moving one, eukaryotic and prokaryotic. Also I added an image of 5 kingdom classification put forward by Robert Whittaker, that is Monera, further Protista, plants, animals and fungi. Now I added a chart over here, so which makes it easier to understand. That is from the text 1.1, 5 kingdom classification. Living world is divided into prokaryote and eukaryote. Prokaryotes are further divided into unicellular. So in prokaryote, there are only unicellular species, that is single celled species. Eukaryotes, unicellular and multicellular, single cell and many cell. Unicellular kingdom Monera. Uh, Eukaryotes, unicellular kingdom Protista. So unicellular species are present in prokaryotes as well as eukaryotes, and multicellular only in eukaryotes, which is kingdom fungi, plantae and animalia. I added few points, that is cell wall is present, but organelles cannot perform photosynthesis because fungi does not have chloroplast, so they cannot carry out photosynthesis. Plantae, cell wall is present and organism can perform photosynthesis. Animalia, no cell wall is present. Further, kingdom Monera. All type of bacteria and blue-green algae are included in kingdom Monera. So when you say blue-green algae, that is they have got chloroplast, so they can carry out photosynthesis, that is autotrophic organism. Characters. All the organisms are unicellular, means all the species coming under Monera, they are single-celled. They may be autotrophic or heterotrophic. Now as we just read blue-green algae means they got chloroplast. So most of the species, they are autotrophic. They can make their own food by photosynthesis. But there are some species which are heterotrophic. Third point, these are prokaryotic cells without distinct nucleus or cell organelle. That is, the cells, they don't have proper nucleus or clear nucleus, or as you use the term distinct, and various cell organelles are absent. So here students, I added the images of various organisms which come under Monera. You can see a Streptococcus pneumonia which causes pneumonia, Treponema, Vibrio, cholerae, Salmonella, typhi which causes typhoid, Clostridium, Legionella, Staphylococcus. Here student, the second image which I added, which shows the labeled parts of the species, that is cytoplasm, ribosomes, nucleoid, as they don't have nucleus, plasma membrane, cell wall, flagellum which is the tail, helps in locomotion, capsule which protects them, pilus, small hair-like body which is similar to cilia. Now students, for your understanding, I added few animation images of Monera. So you can see the bacteria species here, bacteria or different species coming under Monera. Here you can see how they are dividing by simple binary fission from 1 to 10 to 100 to millions. Here again you can see some more images of bacteria, and here how the species of Monera moving around with the help of flagella, and here again bacteria species. Now coming to the second kingdom, Protista. The first characteristic, Protista are single-celled organism with well-defined nucleus enclosed in a nuclear membrane. Means unlike Monera, in Protista the nucleus is very well defined, that is having nuclear membrane. They have pseudopodia or hair-like cilia or whip-like flagella for locomotion. Means they've got various locomotory organs, cilia, flagella or pseudopodia in case of amoeba. Autotrophs, that is they prepare their own food. Example, Euglena, Volvox, both contain chloroplast. Heterotrophs, Amoeba, Paramecium. So in Protista, there is autotrophic organism as well as heterotrophic. So here again, the figure 1.3 shows Volvox, Paramecium, Amoeba, Euglena. I added one more image of eukaryotic unicellular species right from slime mold, Amoeba, Euglena, Dinoflagellae, Paramecium, diatom and macroalgae. Now again students, I added animation images for your understanding how these protista move. So you can see here various images how the movement taking place inside the body of this species also, and how these microorganisms feeding on much more smaller organisms. See here different species how they are moving and locomoting. Now, for the institution at work, National Institute of Virology, Pune is involved in research on viruses. The institute has been founded in 1952 under the jurisdiction of Indian Council of Medical Research. Now, students, this National Institute of Virology, which is in Pune, they carry out research on viruses. Now, if I give a small example, in the present pandemic, that is corona, the vaccines are made in this institute, that is National Institute of Virology, and they carry out various other research work on viruses. Further, third kingdom, fungi. These are non-green eukaryotic heterotrophic organisms. Most of them are saprophytes. They feed upon decaying organic matter. Now, in fungi, all are non-green, means they don't have any chloroplast. They are eukaryotic, means the cell is very well present with all their components, all the organelles. Heterotrophic, they depend on other organisms. They're saprophytic, that is they feed on dead and decaying matter. Third point, their cell wall is made up of tough and complex sugar molecule, that is chitin. Some fungi are thread-like and many nuclei are present in the cytoplasm. Now, fungi species, again there are microspecies and macrospecies coming under fungi. Now, if I say macro, the best example being mushroom, which everyone knows, and some are edible, means some can be consumed as food as well. And there are smallest known one or thread-like species. Examples: baker's yeast, Aspergillus, which are the fungus present on corn, Paramecium, and mushrooms. Again, the image I added over here of fungi, which shows the label part. That is columella, sporangium, apophysis, hypa, sporangiospore, and here this sporangiospore releasing the spores, and each spore forming a new fungi species. That is why in our surrounding there are many spores of fungus present. But if our immune system is weak, then it can cause fungal infection. Here I added again few images of various fungi species right from mushroom to different species in fungi. Here you can see bread mold. That is, if bread, if it is kept out in open or if it crosses the expiry date, you can see a white layer forming, which is the fungus formed on the bread, which is bread mold. Now, classification of microbes. Among the living organisms, microorganisms are largest in number. Hence, they are classified as follows. Now, students, as we learned, there are 87 million species till 2011. That means now it might be much more. It might have crossed 100 million, right? So, to learn these many species, it is necessary to learn and classify. So, here microorganisms are classified as prokaryotes and eukaryotes, as we learned this earlier. Further, prokaryotes are divided into bacteria, whereas eukaryotes into protista, fungi, and algae. And you can see the size, which may vary from 10 raised to 6 micrometer to 10 raised to 9 nanometer. Here I added another image showing the bacteria, fungi, protista, algae, including the viruses, because viruses are also coming under microbes. Now, first one, bacteria. So you can see here the size of a bacteria is 1 micrometer to 10 micrometer. So minute. First point, unicellular independent parasitic organism. Sometimes many bacteria forms colony. Means the bacteria, they are unicellular, means they have got single cell. They are independent and they cause infection. There are organisms which can cause infection, parasitic. And some bacteria, they can form colonies. Like if you see here, the animation I shown here, the bacteria, they're dividing by binary fission and they're forming a colony. They reproduce by simple binary fission, but they don't have any proper nucleus or cell organelles present. In favorable condition, bacteria grow vigorously and can double their number in 20 minutes. So you can see in this particular slide, how fast they are multiplying and growing. On towards left side, you can see another image of a bacteria having a flagella. Moving to the next slide, that is protozoa. Now protozoans are approximately 200 micrometer. First point, protozoans are found in soil, freshwater and seawater. Some are found in the body of other organisms and are pathogenic. Now protozoans are again found everywhere, whether it is water bodies, salt water or fresh water, or even in the soil. Now some can be pathogenic, means causing harm to the different animals or humans. These are unicellular organisms with eukaryotic cell. Now students, like Monera, even unicellular species are present in protozoa, but they are much more developed in comparison to the Monera with the various cell organelles. There is a great variation in cell structure, organs of locomotion and modes of nutrition among protozoans. Again, as we learned earlier, the locomotion can be done by cilia, flagella or even by pseudopodia in case of amoeba. These organisms reproduce by simple cell division. Example, amoeba, paramecium, which are present in free living in dirty water. Entamoeba histolytica, which causes amoebiosis. As suppose you are drinking water from outside, if it is contaminated with this particular species, then it can cause vomiting, stomach pain, diarrhea, loose motion, etc. Plasmodium vivax, which causes malaria. Now, students, as we know, mosquito, that is female Anopheles mosquito, if it bites causes malaria because it carries this particular Plasmodium protozoan in its stomach. When it bites or it injects the Plasmodium into the host organism while sucking the blood. Third, Euglena-autotrophic. Why they are autotrophic? Because they are green in color, having chloroplast. So students, you can see this animation image of Euglena, which is green in color because of chloroplast. They got flagella for locomotion. Here you can see the animation image where mosquito is injecting and plasmodium is getting transmitted into the human body. And here movement of Entamoeba histolytica. Moving further to the third type, fungi. The size is approximately 10 micrometer to 100 micrometer. First point, these are found on decaying organic matter and dead bodies of plants and animals. The fungi species are mostly saprophytic, as again we have learned earlier, means they feed on dead and decaying organic matter. These are eukaryotic organisms. Some are unicellular and some are visible with naked eyes. So again, in fungi species, there are unicellular species which are very minute, and there are multicellular species, bigger species like mushroom. Also, they are eukaryotic, but they don't have any chlorophyll, so they are not photosynthetic or autotrophic. That's why they are dependent. So they are saprophytic. Saprophytic absorb their food from decaying organic matter, means dead and decaying matter. They reproduce sexually and asexually by cell division or budding. That is, sexually by gametes, male and female gametes, asexually by budding. Now, if you see here, students, again I added animation images. Now, if you see in dove, we add uh, yeast or keep it for fermentation, so it starts to ferment. You can see yeast how they are multiplying by budding. Also, you can see another animation image over here. Here animation image of the mushroom. So you can see the yeast how they are multiplying, the candida species how they are growing, and the mushroom. Next, algae. Size is approximately 10 micrometer to 100 micrometer. They are aquatic, that is, they are mostly found in water bodies, which can be fresh water or salt water. Second point, eukaryotic unicellular autotrophic organism. Eukaryotic, now as all of you know, well developed cell, nucleus with all these cell organelles. Unicellular, they are single cell. Autotrophic, they prepare their own food with the help of chloroplast using photosynthesis. The third point, photosynthesis is carried out with the help of chloroplast present in the cell. Example, chlorella chlamydomonas. Fifth point, very few species of algae are unicellular. So as we just read the example, chlorella chlamydomonas are unicellular. So few species present in algae are unicellular. Rest all these species are multicellular. Even they have huge species of 60 feet long in the water body. If I give an example, it's called a sargassum, which is very huge. Most of them are multicellular and visible with naked eyes. Now, coming to the fifth type, viruses, which are approximately 10 micrometer to 100 micrometer, means very small, minute, which are not visible with our naked eyes and not even in the simple or compound microscope, but rather in an electron microscope. Generally, viruses are not considered as living organism, or they are said to be organism at the edge of the living and non-living. They are studied under microbiology. Now students, why this term is used, organism at the edge of living and non-living? Because if the virus, they are outside of any living organism, the living can be plant, animals or humans. So if they're outside the body, they are as good as stone. Now what does stone do? It remains in one position. Same way viruses, they are non-living if they are present outside the body. But once they enter into any living organism, whether it is a human, animal or a plant, they can cause severe infection. Okay, that's why they are also studied under microbiology, that is microorganisms. The first point, viruses are extremely minute. They are 10 to 100 times smaller than bacteria and can be seen only with electron microscope. Now students, you can imagine bacteria itself is very small, which are not visible with our naked eyes. We have to use microscope, and viruses are 10 to 100 times smaller than a bacteria. So these viruses are such that they can even infect a bacteria, okay, or cause harm to the bacteria. And the viruses can only be seen under a electron microscope because they are very minute. They are found in the form of independent particles. Viruses is a long molecule of DNA, that is deoxyribonucleic acid, or RNA ribonucleic acid, covered by a protein coat. So because of this protein coat, the viruses are not easily affected by any antibiotic or vaccines or medicines. Third point, viruses survive only in living plant or animal cells and produce their own proteins with help of host cell and create their numerous replica. Then they destroy the host cell and become free. These free viruses again infect new cells. Now students, as again you might have seen this present pandemic, they are spreading like a wildfire, right? Now these viruses, once they enter into a human body or plant or animals, they start to replicate. They have got their own proteins, but they use the cell or the nucleus of the host organism, the plant or the animals. So they enter the cell or the nucleus of the host organism and use that nucleus to make a replica or copies of it. If I give a small example, when you go and take Xerox copies of your notes, you may take 1 copy, 5 copy, 10 copy. When you put it in the Xerox machine, the copies are print as many numbers you want. Same way these viruses, once they enter into our human body, they make copies using the machine of our body or the plant or the animal body, that is the cell and the nucleus. And once they multiply, they come out of the body and they infect other cells, or they start spreading out to other cells and other organisms. Viruses can cause many disease to plants and animals. So viruses can infect all the living organism, whether it's plant, animal, humans, and even bacteria. And they can cause various disease depending on the type of virus, because there are hundreds of viruses present. Here you can see simple animation of a virus which I have added over here. You can see there's a projection present on the surface of the virus. These are the protein molecules present which are of different shapes in different viruses. Hence all the viruses are different present on this outer covering. Here you can see how they infected the tomato plant. So the name tomato spotted wilt virus. You can see the coloration of the tomato after the infection of the virus. Moving further, do you know? That is, as I mentioned, the virus can infect all the living organisms, right from humans, plants, animals. So here in case of human, polio virus, influenza virus, HIV, AIDS virus, etc. Whereas in cattle, that is cow, buffalo, it is picorna virus. Plants, tomato, wilt virus, as you just saw the image in the earlier slide, tobacco mosaic virus, or in short TMV. Bacteria, as I said, bacteria itself is very small, minute, which are not visible to our eyes. Even they are infected by the viruses, by a virus called as bacteriophage virus. Here students, I added the images of tomato spotted wilt virus, polio virus, influenza virus, HIV, that is human immunodeficiency virus, picorna virus, which causes infection in the cattle. Here I have shown the structure of TMV, tobacco mosaic virus. So you can see students how complicated the structure within inside the cells of the virus are. Bacteriophage, how the structure looks like. Again I added a labeled image and the animation one. The animation one, you can see how this Virus, bacteriophage, comes onto the surface of the body, injects their DNA or RNA into the host organism and infecting the plant or the animal. Okay. Lastly, I hope all this animation and the pictures help you to understand the concept more clearly. The video will be helpful for you for revision. So thank you, students.